KREBS cycle is also called as TCA cycle or citric acid cycle. It is the oxidation of acetyl-CoA to carbon dioxide and water. It is the final common oxidative pathway for metabolism of carbohydrates, fat, and amino acids. It occurs in the mitochondria. It occurs in aerobic conditions only, and it uses 2/3 of oxygen consumption of the body. The pyruvate produced at the end of glycolysis is converted to acetyl-CoA by a process of oxidative decarboxylation. This reaction is catalyzed by a multi-enzyme complex called pyruvate dehydrogenase complex. This enzyme is present particularly in the mitochondria, and it is dependent on 5 cofactors, which is TPP, which contains thiamine, also called as vitamin B1, NAD+, FAD, lipoic acid, and coenzyme A. Oxidative decarboxylation of pyruvate to acetyl-CoA is a connecting link between glycolysis and the TCA cycle. In this process, CO2 is released and therefore acetyl-CoA becomes a 2-carbon compound. Energy is also released in the form of NADH. Now, the TCA cycle begins with the condensation of this 2-carbon acetyl-CoA and a 4-carbon oxaloacetate to form a tricarboxylic acid, which is called as citrate. This occurs by an enzyme citrate synthase. Now, citrate is isomerized to isocitrate by the enzyme aconitase. In this process, an intermediate product is formed which is called as cis-aconitate. Isocitrate is converted to oxalosuccinate, which is converted to alpha-ketoglutarate by the enzyme isocitrate dehydrogenase. In this step, energy is released in the form of NADH along with carbon dioxide. Therefore, alpha-ketoglutarate becomes a 5-carbon compound. Alpha-ketoglutarate is converted to succinyl-CoA through oxidative decarboxylation. In this reaction, again energy is released in the form of NADH along with CO2, and it is catalyzed by alpha-ketoglutarate dehydrogenase. So succinyl-CoA becomes a 4-carbon compound which is converted to succinate by succinate thiokinase. This is an example of substrate level phosphorylation that occurs either by phosphorylation of GDP to GTP in liver and kidneys and ADP to ATP in brain and heart. So depending upon the location of where the citric acid cycle is taking place, the phosphorylation is different. Succinate is oxidized by succinate dehydrogenase to form fumarate. In this process, FADH is released. FADH is equivalent to 1.5 ATPs. Fumarate is converted to malate by fumarase. Malate is oxidized to oxaloacetate by malate dehydrogenase. Again, NADH is released. This oxaloacetate is regenerated, can combine with another molecule of acetyl-CoA, and the TCA cycle continues. Now let's look at the energetics in KREBS cycle. The first enzyme, isocitrate dehydrogenase, produces 2 NADH, which is equivalent to 5 ATP. Alpha-ketoglutarate dehydrogenase, 2 NADH, again 5 ATPs. Succinate thiokinase produces 2 ATP. Succinate dehydrogenase produces 2 FADH, which is equivalent to 1.5 into 2, 3 ATPs. Malate dehydrogenase, 2 NADH, which is equivalent to 5 ATPs. So that means KREBS cycle produces a total of 20 ATPs. Pyruvate dehydrogenase complex was also producing 2 NADH, therefore 5 ATPs from that. And glycolysis was producing 7 ATPs. So that is the total energetics from one molecule of glucose until its complete oxidation to carbon dioxide and water.